Today we are going to talk about an innovative use of infrared spectroscope. Infrared spectroscopy is a non-invasive technique used in early cancer detection. It involves the measurement of the absorption, transmission, and reflection of infrared light by biomolecules in biological tissues. Cancer cells exhibit distinct spectral fingerprints due to their unique biochemical composition. By analyzing these spectral patterns, infrared spectroscopy can identify subtle molecular changes associated with cancer at an early stage. This enables timely diagnosis and intervention, improving the chances of successful treatment. Infrared spectroscopy offers a promising avenue for precise and minimally invasive cancer screening, contributing to early detection and improved patient outcomes. Firstly, let's check how early detection of cancer using blood testing is achieved. We know cancer cells multiply and spread indefinitely in the body, as well as releasing a variety of abnormal substances such as metalloproteinases and collagenases. These abnormal substances can destroy surrounding tissues and basement membranes. After the barrier is destroyed, cancer cells will penetrate through blood vessel and lymphatic vessel walls, entering surrounding tissues and lymph nodes, forming metastatic lesions. To fight against cancer, our body also produces some other substances, such as interferon, which is a type of protein molecule that can stimulate the immune system, enhance the body's ability to attack cancer cells, and inhibit the growth and spread of cancer cells. We can identify the presence of cancer by detecting these substances in our circulating systems. Now, we can use the method of cell biopsy to extract and quantify ctDNA and cfDNA in our blood. This will help determine the presence of cancer cells, the organ of cancer origin, and the progression of the cancer. However, the current data shows that this type of method has a low accuracy rate, with only a 75% detection rate for ctDNA in advanced cancer. Additionally, not all cancer cells release gene information, especially in the early stages of cancer, which makes it very hard to detect cancer earlier. So let's talk about the second method, in which we can detect abnormal substances and other substances in human blood, which broadly fall into 7 categories: embryonic antigen, glycogen, hormones, proteins, enzymes, receptors, and genes. These 7 categories can reflect the occurrence of most tumors. Let's use one example to explain how this works. We know many proteins are only secreted during the embryonic period and stop being produced after birth. When tumors occur, the tumor cells will resynthesize and secrete embryonic-specific proteins, which we call embryonic antigens. This type of biomarkers include alpha-fetoprotein for primary liver cancer and carcinoembryonic antigen for common digestive system tumors and others. For example, when normal cells transform into malignant cells, the glycoproteins on the surface of the cells mutate and form special antigens, which are called glycogens. This type of biomarkers include CA50 for pancreatic, colon, and rectal cancer, as well as CA125 for ovarian epithelial cancer. So how are these substances detected? Traditional tumor screening uses enzyme-linked immunosorbent assay ELISA or chemiluminescent immunoassay. ELISA can only detect a single indicator in one test. It is slow and costly. Therefore, it has been gradually replaced by CLIA. CLIA takes 2 hours to determine biomarkers and costs $100 to $300 per time. The challenge for these 2 traditional methods is that they can only test one different type of substance or DNA at a time. During the early cancer screening, we don't have any idea if a person may potentially have which cancer. You have to run a lot of tests to find out if this person has a cancer. So, can we test all of the cancers at the same time? Yes, we can with this new technology called infrared spectroscopy examination. And the most advantage of using infrared spectroscopy is that it does not limit it to specific cancer target, as it can detect different types of tumor cells and different types of substances produced by tumors in the blood. At the same time, it's an amazing technology. And let's talk about how it works and how it is used to detect cancers. First, we have to understand the principle of infrared spectroscopy. Infrared is a type of electromagnetic radiation. According to its wavelength, there are radio waves, infrared, microwaves, visible light, ultraviolet light, X-rays, and gamma rays. The radio waves has the longest wavelength, and the gamma ray has the shortest wavelength. So infrared has longer wavelengths than visible light and shorter wavelengths than microwaves. It is commonly referred to as heat radiation because it is mostly emitted by warm objects and can be detected as heat by the human body. Infrared is used in a variety of applications, including thermal imaging, remote sensing, and communication. Infrared spectroscopy is used to analyze the structure and properties of chemical compounds and how it works. When infrared passes through a crystal with a high refractive index and is then projected onto the surface of the measured materials, here is the measured material. In this video, we call them samples. Refractive index is a measurement parameter of how much a material can refract light as it passes through it. The refractive index of a material depends on its composition, density, and other physical properties. The higher the refractive index of a material, the more it can bend light as it passes through it. For example, diamond has a very high refractive index, which is why it is so sparkly and reflective. On the other hand, air has a refractive index very close to 1, meaning that it doesn't significantly alter the path of light passing through it. Refractive index is an important concept in optics and is used to understand the behavior of light as it passes through lenses, prisms, and other optical components. In our case, not all of the infrared light is reflected back, but rather it penetrates into the surface of the sample to a certain depth before returning to the surface. During this process, the sample absorbs infrared light, resulting in a reduction in the intensity of the reflected light. The scale of the reduction depends on the structure of the sample. For example, if the sample is water, we use water to explain how it works, because water has a very simple structure, and we know it very well. So with the infrared spectroscopy, we can get a very simple wave pattern. There are 2 peaks that reflects the structure of water molecules. Let's use another example. Let's change the water to diamond as a sample, because diamond also has a very simple structure. With the infrared spectroscopy, we got a light absorption curve, or we call it spectrum. With these 2 examples, we understand when the light is passed to the sample, the absorption light spectrum has different pattern for different molecular structure in sample material. Now let's look at spectrum of samples contains cancer gene or substance we mentioned earlier. This is a comparison chart between brain cancer patients and non-brain cancer patients. This is a comparison chart between kidney cancer patients and non-kidney cancer patients. The entire sample's characteristics, including cell-free tumor markers, tumor methylation markers, exosomes, proteins, and carbohydrate metabolites, are reflected in these spectrums. Based on this complete spectrum, and by comparing it with an existing database, artificial intelligence algorithms can provide results within minutes and show the possibility of the user having cancer. As you can see, it can be difficult to make judgments with the naked eye. That's why some devices also incorporate artificial intelligence algorithms to improve accuracy.